Hello, and welcome back to your online lectures. In this chapter, chapter 9, we're going to be focusing on cellular respiration and fermentation. And even if you don't know exactly what those are, you kind of already do because you know that we need to eat in order to make energy for ourselves and that we need oxygen. And cellular respiration and fermentation are going to take all of these concepts and put them together for you. Except we're going to be going into the molecular aspect of it. So you're going to actually see, we always say that mitochondria are the powerhouse of the cell. So you're going to actually see how and why we call it that. And you're also going to see why we need oxygen. Because a lot of people know we need oxygen, but you don't quite know what it's used for. So you're going to see how we utilize oxygen, how we break down food, particularly glucose, and how we derive energy with the mitochondria, which does our cellular respiration. In this chapter, students will be able to explain the processes of energy transformation as they relate to cellular metabolism. Particularly, you'll be able to describe both molecular and energetic input and output for cellular respiration. You'll do some model or mapping of the cellular organization of metabolic processes, and you'll be able to model or map the consequences of anaerobic and aerobic conditions to cellular respiration. So as I mentioned, cellular respiration, the goal is to create energy. And we learned in the last chapter, in chapter 8, that energy is the capacity to cause change or to do work. And there are 3 types of work that we learned that our cell can do in the body, and they are chemical work, transport work, and mechanical work. Chemical work, for example, would be chemical reactions, the making and breaking of bonds. Transport work would be moving things across a membrane, particularly active transport. In chapter 7, we learned the difference between passive transport and active transport. Passive is diffusion. It uses a concentration gradient to move things across the membrane, so no energy is needed for passive transport. But active transport does need ATP because we are going against concentration gradients. And then mechanical work would be, the example that they have here is the contraction of a muscle. Your muscles need a lot of ATP in order for them to contract for you to move. So you can imagine how much energy each one of your cells requires, and you have 1 trillion or more than 1 trillion cells in your body, right? So living cells require energy in order to do all of this work. And where do we get energy from? We have to get it from outside sources. Animals in particular, we obtain energy for our work by feeding on other animals or photosynthetic organisms. Photosynthetic organisms would just be plants, right? So we eat rice and wheat and beans and vegetables. So that's where we get our energy from, so that our cells can do their 3 types of work. So we're going to look at this energy diagram here, and we're going to see that energy flows into an ecosystem as sunlight and leaves as heat. So this is a really important little snapshot of the next 2 chapters that we're going to be learning. This chapter here is cellular respiration, right? So we're going to be focusing on this part of the cycle. In the next chapter, we're going to hit photosynthesis, and we're going to focus on the top. But let's look at photosynthesis. So we know that plants require water and carbon dioxide, right? They're able to take this water and carbon dioxide, and through their chloroplast, they have special mechanism where they can do photosynthesis, and they use the energy from the sun, light energy, in order to turn these reactants into these products. The products are organic molecules and, of course, oxygen, right? We know that we breathe in oxygen from plants. Organic molecules would be the the macromolecules that we learned in chapter 5. So that would be carbohydrates, proteins, and lipids. Those are all carbon-based molecules, which makes them organic molecules. And our photosynthetic organisms are what build them for us. So they release these carbs and proteins and lipids and oxygen. Now, respirators, cellular respirators, like us, we are able to take these organic molecules and oxygen, and through our mitochondria, we are able to utilize these to, number one, produce ATP, which is our source of energy, and we have our byproducts. Of course, you know that we exhale carbon dioxide, and then as you'll see, you may have already started to notice, but water is often involved in our chemical reactions, and it's often a product of it. So we release water, and it's part of our excretion too, right? So energy is flowing into this system via light energy, and then it's leaving this system via the formation of ATP and heat energy. And most of the energy that we take in via our eating, our organic molecules, I think about 60% of it is actually lost as heat. And this is why our bodies are warm. Heat is a byproduct of all of our metabolic reactions along with ATP. And you can feel that if you go near any living organism. As I mentioned in the previous chapter, chapter 8, we learned about the metabolic pathways, our metabolism, which is comprised of all catabolic reactions and all anabolic reactions. And those are just two opposites. Catabolic break things down, anabolic builds things up. And that's really all you can do, right? You can either make or break molecules in your body. So we are going to be focusing on the catabolic pathways because if you also recall, we learned about the different types of energy, and we learned kinetic energy, which is the energy of motion, and we learned potential energy, which has to do with either location or structure. And the example that was shown in your slides, so this is it here, for potential energy is chemical energy. Chemicals meaning molecules. So an example of potential energy is the energy that's stored in the bonds of molecules. So of course, if we break them apart, we're going to be able to start harnessing that energy. So that's catabolic pathways. That's catabolism. Particularly, we are going to be talking about breaking down glucose molecules to harness energy. So that's why we're talking about catabolic pathways here. We're going to be looking at the electron transfers once we start breaking down our glucose molecule, because this is really important. This is actually how we're going to be building ATP. Just imagine we have a large molecule, like a glucose molecule, and we start breaking it down. We're kind of ripping it apart. And in doing so, this is called oxidizing, by the way, when we break down molecules. In doing so, we can start collecting these electrons, and we can take these electrons and we're going to shuttle them somewhere, and they're going to help power the creation of ATP. So let's look at that a little bit. So you really can't have biology without chemistry, right? So we do have a little bit of chemistry here. Hopefully you've heard of redox reactions, but if not, we're going to go over them. So I just mentioned oxidation, so we're going to learn about that. We know that atoms have 3 subatomic particles, right? They have neutrons and protons in their nucleus, and then orbiting the nucleus are the electrons, which are the smallest of the subatomic particles. They have very little mass. They're zipping all around, and they are what are involved in all of the chemical reactions. Look back in chapter 2 if you're confused about electrons, because it's important to understand that chapter as a foundation. We know that whether electrons are stolen or shared determines the different types of bonds, right? So electrons are really important in just how a molecule is functioning. So let's just break it down even simpler. If an atom, or sorry, if a molecule can either gain electrons or have them stolen. And that's what these two words, oxidation and reduction, are. In oxidation, a substance loses electrons, and in reduction, a substance gains electrons. It's that simple. So if something is oxidized, it has lost electrons. And I just kind of mentioned that in the previous slide, where we're talking about the breakdown of glucose, and we're going to be yielding energy by oxidizing organic fuels, by breaking them apart, the glucose, and they're going to be losing electrons. So oxidation is the loss of electrons, and that molecule will then be quote unquote oxidized. Reduction is the opposite. If you reduce something, you're giving it electrons. That's simple. And also, just remember that electrons are charged with a negative charge. So if we're ripping electrons away and adding electrons, we're also altering the charges of these molecules. So just keep that in mind. So then, if oxidation is the loss of electrons and reduction is the gain of electrons, what's a redox? Well, redox is reduction and oxidation, right? It's those two words put together, redox. Because if one molecule has an electron and loses it, and the other one gains it, both of those things were occurring. One of those molecules lost the electron while the other one gained it, right? Just imagine somebody passing a ball back and forth between each other, and the ball is the electron. Someone starts with the ball, they lose that ball, they were oxidized, and then the person that gained that ball would be reduced. They gained the ball, which is our electron here. So essentially, both are occurring at the same time. So that's why these are called redox reactions. They're always both occurring. So look to this very basic reaction shown here. We have X with an electron, right? They're negatively charged. So that's the E minus. So we have X with an electron plus Y. So these are just two chemical reactants that are near each other. They're going to be part of a chemical reaction, and they go through the reaction, and the products, what's produced, we have X, and it's lost its electron because the electron was transferred to Y. So now we have Y with the electron as the product. So X started off with the electron and lost it. So X was oxidized. Y started off without the electron but gained it, right? So Y becomes reduced. Now you'll also see here that the one that becomes oxidized, the one that loses the electron, is what's reducing the other one, right? It's causing X is causing the Y to gain an electron, to be reduced. So X is the reducing agent. So basically, if X is being oxidized, meaning it's losing its electron, it is the reducing agent. And then if Y is becoming reduced by gaining the electron, it is the oxidizing agent, because to oxidize means to lose an electron, so it is making X lose its electron, right? So just very basic. X starts with the electron, gives it over to Y. X lost it, so it was oxidized. Y gained it, so it was reduced. The one that is oxidized is the reducing agent, and the one that is reduced is the oxidizing agent. So these are just a very basic formula here. We're going to apply this to cellular respiration. So here we go. This formula should look a little familiar. C6H12O6 plus 6O2, that's oxygen. So this is a glucose plus 6 oxygen molecules yields 6 carbon dioxide, 6 CO2, and 6 waters, 6 H2O. This is the formula for cellular respiration, and make sure that you know this formula. And just as a precursor for the next chapter, photosynthesis is the same thing in reverse, right? And then we also have energy here because energy is also released, right? We're making ATP because of all of this. So it's the same thing that we just saw, except now we replace the X and the Y with our actual molecules that we our reactants that we start with and our products. So here is our glucose, right? We've eaten and now our body has broken down all of that food into the basic monomers. So this is a monosaccharide of glucose. So a glucose monosaccharide, we are going to be breaking this down into its components. So remember, when we break things down with catabolic reactions, that's oxidizing. So glucose becomes oxidized to carbon dioxide. So where do all of these electrons go? Because to oxidize means to lose electrons. Well, they're going to be going on to the oxygen. So the oxygen, the O2, is becoming reduced, and the result is that it's also going to gain a couple of hydrogens. We'll see how that happens, but it becomes water. So this is what we're breathing in, this is what we're eating. Goes through our mitochondria, we release carbon dioxide, and we release water, and then we create energy. So essentially what we've just seen is an electron transfer, right? The electrons begin on our organic molecule, which is glucose, and through a bunch of metabolic processes. I know this is a very simplified, streamlined version. It's not just as simple as the electrons shuffle over to here. We're going to step through it, but there's, I don't know, maybe 30 or so reactions in between all of this. So this is just a very complex system of electron transfer whereby they get ripped off, oxidized from our glucose, and eventually get added. The electrons get added onto the oxygen, and we create water and carbon dioxide. So we're going to see more about the electron transport chain a little bit later in the lecture, but we are going to just cover one aspect of it here, and just to kind of cover it, and then when we get back to it, I'll go over it again. But so I mentioned that there's, I don't know, maybe 30 or so steps in between all of this, right? So let's say that once we rip all of the electrons off of here, well, where do they go, right? So there's actually a special molecule that's part of this process. It's an electron shuttler, right? So its job is to grab the excess electrons that are being ripped off from the molecule as it gets oxidized, and it gets shuttled to a certain spot. So this is that molecule. It's called NAD+. NAD+ is a coenzyme, and we also will say that it's an electron acceptor. It functions as an oxidizing agent during cellular respiration. So let's see here. Here is the picture of NAD+. NAD stands for nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide. But you just have to know NAD. It has a plus charge on it, so you can see already that it will have an affinity for electrons. So one NAD+ molecule here, the coenzyme, is able to grab onto 2 electrons. We'll also notice that when molecules grab these electrons, again, electrons are negatively charged, And we don't want negatively charged molecules in our body just floating around, right? So in addition to grabbing 2 electrons, they tend to also grab the opposite of the negative charged thing, which is a positively charged proton, to neutralize that charge. So it'll grab 2 negatively charged electrons and 2 positively charged protons, so that this doesn't have an additional charge on it. So this is just that reaction here. 1 NAD plus 2 hydrogens, and then the 2 electrons here, we make an NADH. So NAD plus 2 hydrogens plus 2 electrons makes an NADH. And now this is the reduced form of nicotinamide, and this is carrying the electrons. It's an electron shuttle, and we'll see later on where all the shuttles take the electrons to make ATP. But for now, just take note of NAD as the electron transporter. So now let's actually get into cellular respiration. So there are 3 main stages of cellular respiration, and they are glycolysis, the citric acid cycle, or you may have heard of the Krebs cycle, and oxidative phosphorylation. So we're going to go into each one, break them down. Here is a nice overall snapshot of those 3 stages. We have glycolysis, the citric acid cycle, and oxidative phosphorylation. So you can tell that they're all distinct. They all have a different place that they occur. They have different reactants going into them, different products that they're creating, and they're a chain, right? So we have the first step, and then we go into, this is kind of like a half step here, and we'll go over that. Then we go into the second step, and then we go into the final step. And we can also see that there's a little bit of ATP being made at each step. So while we go through these, I'm going to be calling out a couple of important factors that you need to note and pay attention to. Number 1 will be the reactants, so what we begin with. Number 2 will be the products, so what we end with, right? Number 3 will be the ATP that's made, and number 4 is going to be the shuttles, the NADH, and there's actually another one here called FADH2. We'll get to that in a second, but they both function in the same way. Those are our electron acceptors that are going to be shuttling the electrons, and we shuttle them to the very end over here. So when we get to there, you'll see what happens. But that's what you need to pay attention to, as well as the location. So step 1 is glycolysis. Glycolysis means sugar splitting. So that should be giving you an indication right there. Anytime you hear glyco or gluco, right, you should think of sugar. So glycolysis, lysis means to split, right? Glycolysis, glycolysis, sugar splitting. So that tells you we are going to be starting off with a glucose monosaccharide and breaking it down. We're going to start oxidizing it. Couple things to note about glycolysis. The location. Glycolysis always occurs in the cytoplasm. So we see here the mitochondria, which is our ATP producer, right? Glycolysis is occurring outside of the mitochondria. So I also want to point out that there's no organelle involved in this step. So what does that say about eukaryotic and prokaryotic cells? Obviously, this is a eukaryotic cell, right? We are eukaryotes, so we have organelles. Glycolysis occurs in our cells as part 1 of our cellular respiration, but glycolysis is a very old mechanism, and glycolysis actually also occurs in bacteria. So because there's no organelle required, so it's occurring in their cytoplasm and their cytosol. So just note that. And we're going to go over fermentation in a little bit. So this is just a little bit precursor to that. But glycolysis occurs in the cytoplasm, and there are two major phases of it. We have an energy investment phase and an energy payoff phase. So let's look. And then one other thing, it occurs whether or not oxygen is present. I'll double back to this concept when we see where oxygen comes in, because I think right now it doesn't really mean much to you. So here are our two stages of glycolysis. We have the energy investment phase and the energy payoff phase. I'm going to go over this very basically, and then in the next slide, I'll show you the actual reactions that are occurring so that, if you're needing to know that, you can see that too. So what do we mean by energy investment? Well, we have to pay a little bit of energy to get a little bit of energy. So we begin with glucose for glycolysis, right? So this is our reactant. We begin with glucose. And what happens is we actually have to put in 2 ATPs. We have to pay a little bit in order to get this reaction going. So that's why this is called the energy investment phase. So we start off with glucose. We have to pay 2 ATP. What happens when we start off with ATP? Remember from the last chapter that ATP, when we break off the third phosphate group, the hydrolysis, which remember hydrolysis, water breaking. So hydrolysis is when we add in a water to break a covalent bond in a polymer. So the hydrolysis of ATP releases energy. So it's an exergonic reaction. And we're able to harness that energy. So breaking this ATP here, we have actually we'll look at here. So we have the 3 phosphates. We're going to hydrolyze this last one here. So we're left with an ADP, which is an adenosine diphosphate, right? This is adenosine triphosphate. We break one off. We have adenosine diphosphate. And we have an inorganic phosphate. That's that phosphate group that was on the end here that got cleaved off. Why is it called an inorganic phosphate? Remember, organic chemistry means carbon-based. Well, this phosphate group is no longer attached to a carbon skeleton. The carbons are here. So now this is inorganic phosphate. These phosphates are organic because they're attached to carbon-containing compounds. So we have an ADP and an inorganic phosphate and energy. And that is exactly what we see here. We have our 2 ATPs that we fed in, and we have 2 ADPs released and 2 inorganic phosphates released. Okay, so that's the energy investment phase. The second part is the energy payoff phase. So this is where we get something back. So now we are able to take 4 ADP and 4 inorganic phosphates that are hanging around the cell and put them together, pop them together to create 4 ATP. So this is great. We've created some energy. We also are taking 2 NAD+. Remember, those are our electron shuttles. And for each NAD+, we're going to use 2 electrons and 2 hydrogen. So we need total 4 electrons and 4 hydrogen, and we're making 2 NADHs. So this is good too. This is going to also help us to make energy a little bit later. And so these are our energy inputs and outputs here. The molecules that we create, our products, we make 2 pyruvates and 2 waters. So looking at the net creation, we started off with glucose and we make 2 pyruvates. We started off, we created 4 ATP here, but we used 2 here. So our net creation of ATP is only 2. It's 4 minus 2. So glycolysis produces 2 ATP per 1 glucose molecule. And altogether, we've created 2 NADHs from glycolysis as well. So one glucose molecule makes a net of 2 ATP and 2 NADH, and this glucose molecule got broken down into 2 molecules of pyruvate. So let's look at this again, but a little bit more in depth. Okay, here it is in depth. So you don't need to know this for an introductory bio class, but I'll just let you know that for biochemistry, you 100% need to know these and probably have to know how to draw them. So it's good to at least start introducing yourself to it and seeing it and kind of getting a general idea, understanding of how glycolysis functions. So here's those 2 phases. We have the energy investment where we have to pay a little bit of ATP, and then we have our energy payoff where we're making, here's 2, and then here's 2 more, 4. So we paid 2, we made 4, so we netted 2 here. So let's look at this. Glucose. Glucose is C6H12O6. Make sure you know that. So glucose, and this is the other thing that I'm going to be pointing out to you, the number of carbons, because it's always good to track the carbons in this cellular respiration cycle and in photosynthesis when we get to it. So glucose is a 6-carbon molecule, okay? Notice, so we go through the first phase, which is 5 chemical reactions, right? Every single one of these numbers here is an enzyme. We can see the enzyme that ends in ACE, ASE. So each one of these is a chemical reaction here. So we have 5, and then we have another 5. So glycolysis is a total of 10 steps. We start off with our 6-carbon glucose, and what do we end with? We end with a 2 3-carbon pyruvates. That makes sense, right? 3 plus 3 is 6. So we start off with 6 carbons, and we have 2 3-carbon molecules, right? Carbon 1, carbon 2, carbon 3, called pyruvate. We are taking apart we're in oxidation, each step, each enzyme does something specific. So as you see in this first step where we're paying the ATP here, what are we doing? Well, what happened? This hexokinase enzyme added on a phosphate. And where do we get the phosphate from? We got it from ATP, right? So we kicked out the ADP, right? We released that. We kept the 1-phosphate, and this enzyme popped it right onto this. So we have a new molecule here, glucose 6-phosphate, right? And then we have a couple of other steps here. Here's that other ATP that we paid, right? Same thing happens here. It's hydrolyzed. We release the ADP. And here we have a second phosphate group that got put on. Here's the first one from the first ATP. Here's the second one. And you see how every single enzyme just does like one little thing, one tweak to this molecule. And enzymes are very specific, right, for their substrate and their function. This next enzyme broke that 6-carbon molecule in half. So we have, this is the beginning of 2 3-carbon molecules here. And you don't have to know the names here, but I'm going to point out this one because you're going to see it in photosynthesis. Glyceraldehyde 3-phosphate, which is G3P. So just, glyceraldehyde 3-phosphate. It would be a good one to note because you're going to see this one in photosynthesis again. So now we have 2 3-carbon molecules here going into this second stage, which is our energy payoff stage. So we have glyceraldehyde 3-phosphate, there it is, right, G3P. G3P goes into this next stage, and it doesn't matter, you don't have to know the details, but what do we see here? Here's an NAD plus electron shuttle coming in, and this enzyme is taking an electron off of this and putting it onto the, 2 electrons actually, onto the NADH. So we've created one. And then again here, this phosphate gets put onto an ATP, so we create an ATP. So this 3, and then again over here, right? We make one more ATP, and this 3 carbon molecule, remember we have 2 of them. So this happens twice. That's why we create 1 NADH, 1 ATP, 1 ATP, and then again 1 NADH, 1 ATP, 1 ATP, for a total of 2 NADHs, 2 ATPs here, and 2 ATPs here, and then we have our 2 3-carbon pyruvates at the very end. So again, make sure you know the reactants, that we paid 2 ATPs in the beginning of glycolysis, that we made 4, which gives us a net of 2, and we created 2 NADHs, and our output product here is 2 3-carbon pyruvates. So here we go. Now we've just completed glycolysis. We started with glucose, we've made 2 pyruvates, we made some NADHs, and we made some ATP. The next step is, remember, we are still in the cytoplasm. So the next step is we have to get those 2 pyruvate molecules into the mitochondria to continue with our cellular respiration process. So we're going to do this by pyruvate oxidation. So let's look at that. So to shuttle the pyruvate into the mitochondria, we actually need, this is a multi-enzyme complex that's required to, it catalyzes these 3 different reactions. So let's look at that. The first one is the oxidation of pyruvate and the release of carbon dioxide. The second one is the reduction of another NAD plus to NADH, and then the combination of the remaining 2 carbon fragment and coenzyme A to form acetyl-CoA. So let's look at those. So here we go. Here's our cytosol, or cytoplasm here, where glycolysis has just occurred. Here is a pyruvate that we've just created. We need to get this pyruvate across this membrane. This is the mitochondria, and this is a double membrane. We need to get it across into the internal part of the mitochondria. So here's our enzyme complex here. We have the 3 steps. The first one is the oxidation of pyruvate. Now remember, oxidation, we're breaking it down. So we're going to lose components and or electrons, right? So pyruvate is a 1, 2, 3 carbon molecule. We are going to oxidize it into a 2 carbon molecule. So what happens to this 3rd carbon, which also has 2 oxygens attached to it? Well, this is a CO2. This is a carbon dioxide molecule in the making here. So this first step, we oxidize the pyruvate and we break off this section and we release it as carbon dioxide. So the carbon dioxide that you're exhaling, where is it coming from? It's coming from the oxidation of molecules like this. And we're going to see this a couple times in this step. This is the first step where we're actually breaking off a carbon and releasing it. So that's the first step there, oxidization of pyruvate. Now we are left with a 2 carbon molecule. Okay. Step 2 is, as this is happening, we have a couple of extra enzymes kicking around here. So the NAD plus coenzyme is able to harness those electrons, take them, accept them, in addition to some hydrogens that are hanging around, and we've created NADH, which is another electron shuttle. Fantastic. And finally, we can't just have this 2 carbon molecule sitting this way, so we have coenzyme A, which is going to be added on to this 2 carbon group here. Just as a little aside, this new molecule that we've created with our 2 carbons and then part of this CoA here, coenzyme A, is called acetyl-CoA or acetyl coenzyme A. So the acetyl, you may think of maybe acetic acid, and that's another 2-carbon acid. So you'll notice that there's some similarities in the roots of naming these molecules. So same with acetone, right? If you look at that. So there are 2 carbons to all of those molecules. So this is acetyl-CoA, acetyl coenzyme A. So also remember that this was just 1 pyruvate, and we created 2 at the end of glycolysis. So the first molecule of pyruvate is going to go through this, release 1 carbon dioxide, we're going to make an NADH and 1 acetyl-CoA, and then the second pyruvate molecule is going to do that again. So in total, we are creating 2 carbon dioxides, 2 NADHs, and 2 acetyl-CoAs. And now we're ready to go into the second step, because this is kind of a half step. The second step, which is the citric acid cycle. Here's this image again, just so you can see where we are, right? We moved through glycolysis. Pyruvate was shuttled into the mitochondria. In the process, we released some carbon dioxide, we created some NADH, and we created 2 acetyl-CoAs. So now acetyl-CoA is what feeds into the citric acid cycle. And here it is, the citric acid cycle, also known as the Krebs cycle. So the citric acid cycle is made up of 8 steps. And once again, we see 1, 2, 3, 4, 5, 6, 7, 8. And at each one of those arrows and those numbers, there is an enzyme that is doing that particular reaction, right? And every enzyme just kind of tweaks, does one thing to the molecule. So you don't need to know all of these for an intro bio class. Once again, if you're in biochemistry, you will definitely need to know most likely not only the substrates, but the enzymes involved. But for bio, let's look at these. So you are starting the cycle, step 1 here, with your acetyl-CoA, which you've just created, right? There's our 2-carbon molecule with a coenzyme A attached to it. That's our acetyl-CoA. So this is a 2-carbon molecule. We are going to be sticking this acetyl-CoA onto another molecule called oxaloacetate. And that one you do need to know. There's 3 of them that you need to know here. Oxaloacetate is a 1, 2, 3, 4 carbon molecule. I also mentioned before to follow the carbons. These are actually color coded for you. Just take a look and see if you can find the pattern, because there's a very methodical way that this occurs. Again, it's a little bit beyond an intro bio course, but you're going to have to know it eventually. Might as well kind of start looking at it. So you have your 2-carbon acetyl-CoA. We are going to connect it to this 4-carbon oxaloacetate. So of course, that makes a 6-carbon molecule, which is called citrate, aka citric acid. So here you go, the citric acid cycle. The very first molecule that you create with the first enzyme is citric acid. And again, it's a 6-carbon molecule. So what happens to citrate that eventually turns it into a 4-carbon oxaloacetate? So obviously, we're going to be losing 2 carbons. Can you see how they're lost? And over here, looks just like what we just saw in the previous steps, but we are going to be losing 2 carbons as carbon dioxide, again, which is what we're going to be exhaling. Notice that every time we lose a carbon dioxide, just like in the last step, we make an NADH. Right? This is a dehydrogenase enzyme, but you don't need to know the enzymes. So, so how many NADHs are made in the citric acid cycle with 1 turn of the citric acid cycle? We make 1, 2, 3 NADHs, And you'll see that there's a second type of shuttle here called FADH2. It's the same concept. FAD is the coenzyme, and then it becomes reduced to FADH2. It also grabs two electrons. So we make 1, 2, 3 NADHs and 1 FADH2 with just one turn, in addition to 1 ATP, right? We see that 1 ATP there. We also lost 2 carbon dioxides, so we lost 2 of our carbons in this turn as well. And don't forget, we get to go through this cycle twice because we created 2 acetyl-CoAs. So we're going to go through it a first time, and then our second acetyl-CoA is going to go through it a second time. So altogether, 1 glucose molecule, which created 2 acetyl-CoAs, 1 glucose molecule gives us 6 NADH, 2 FADH2s, and 2 ATPs from the citric acid cycle. So again, looking at this snapshot, we moved through glycolysis, we brought our pyruvate into the mitochondria and we created acetyl-CoA in the process. We got 2 acetyl-CoAs, which each got a turn in the citric acid cycle. In doing so, we released some carbon dioxide, which isn't shown here. We made a couple ATP, and we made a few more of these NADH and FADH2s. In fact, we made a bunch here, right? We made 6 NADH, 2 FADH2s. We made 2 NADHs from this step. We made 2 NADHs from this step, right? And we've only created a net of 2 ATP here and 2 ATP here. So we've only created 4 ATP total up to this point, but we've amassed a good amount of these electron transporters. So for the final step, we're going to see what we do with these. And this final step is called oxidative phosphorylation. So let's look at that. Okay, so as I mentioned, we've gone through all of these steps. There were 10 steps in glycolysis. We had to transfer something across the membrane. Then there were 8 steps, right? We've already gone through 18, 19, 20 steps in order to get to this point, and we've only made 4 ATP, which is not that much. So what we have also created, though, is the NADH and the FADH2s. So these are what give you your bang for your buck with cellular respiration. We are going to be using those for something called the electron transport chain. And if you've taken biology already, this probably sounds familiar to you. The electron transport chain. We learned about redox reactions, right? Reduction and oxidation. Every time a molecule gives away an electron, that is a redox reaction. 1 lost and 1 gained. The electron transport chain or the electron transfer chain is essentially a chain of redox reactions in which an electron is hopped between different molecules or cytochromes, as we'll see, all the way down an energy gradient. So here's a picture of that. This is the electron transport chain in your mitochondria. And there's not just one. I'll show you where there are in a minute. First, let's just get how they function. So here at the top, we see NADH. So here it is, our electron transport shuttle carrier here. This is not just a picture, this is a graph, right? On the y-axis, we have G, our Gibbs free energy, which was from the last chapter, chapter 8, relative to oxygen, right? So this is kilocalories per mole. So this is free energy, able to do work. We have it 0, 10, 20, 30, 40, 50 here. At the bottom of this is oxygen hanging out here, and it says most electronegative. So what this chain is a chain of molecules that go from least electronegative to subsequently more and more and more and more and more and most electronegative. And remember, electronegativity just means how hungry that molecule is for electrons. So we have molecules that are not as hungry, right? They don't have as high of an affinity for electrons, and then they become more and more and more have higher affinity. And this affinity is an energy relationship, right? It is what is going to be pulling this electron down this this energy gradient to move them through these redox reactions, right? Every time an electron hops from one of these cytochromes to another, one of these molecules to another, that's a redox reaction. So we got a redox and redox and redox. The thing that's pulling it is that the one below it is more electronegative. So it's just going to energetically pull it through this electron transport chain until the very end, when oxygen, which as we know is very electronegative, the most electronegative out of all of these, is going to be at the very bottom to catch that electron. So the NADHs and the FADH2s, their job was to grab the electrons as they were being ripped off of the glucose molecule. As glucose was being oxidized, NAD plus and FAD grabbed those electrons to become reduced, shuttled them into the mitochondria and to the electron transport chain so that this electron transport chain could occur. So we're gonna see what happens with this. Here's one more representation of that energy gradient that I just showed you, that free energy. So it's the same thing here, but here's your electron transport chain, and our free energy is increasing over here. What it's trying to show you is a comparison between an uncontrolled reaction and the cellular respiration process. And the reaction being oxygen, this is oxygen gas, right, O2, and hydrogen coming together to form water, right, which is the overall reaction that we just saw. Oxygen was waiting at the bottom to accept electrons, and also anytime it accepts electrons, it wants to accept protons to balance out that charge. But the energy difference between these two is apparently major. And if it were to just occur, there would be just an explosive release of energy. So instead, the cell has these incremental steps of the electron transport chain where each and every one releases a little bit of energy, and it's not just lost. We'll see what it's doing in the process to help create this ATP at the end, because this isn't making ATP, this is just energy, right? I haven't even brought up how ATP is made. Of course, we need to harness energy, so this is an energy flow here. It would be too intense if it was just happen in one step. So our cell has these incremental steps of the electron transport chain here in order to not have an explosive release of energy like that. One thing that you have to note about these proteins here, so there are 4 major complexes, as they're called, and they're called complex 1, 2, 3, and 4, and they're giant complexes of proteins. And then we have a couple of other smaller significant proteins, pieces of this pathway. And they're a special type of protein called a cytochrome. So here's cytochrome. So the electrons are passed through several proteins, including some cytochromes. And cytochromes are just a group of proteins that aid in energy transfer by undergoing redox reactions, right? And that's exactly what's occurring here. They are helping with the transfer of the energy. This electron is moving down its energy gradient, and it's doing this via redox reactions. So just note that these are complexes 1, 2, 3, and 4, and that the proteins that are part of it here are called cytochromes. And that's why you see CYTB, that's cytochrome B, cytochrome C1, cytochrome C, cytochrome A. And this is a picture of a general cytochrome here, and you'll see that there's an iron in the center, and this is called a heme also, and it looks just like the heme group that's in your red blood cells, and as we'll see, it looks like the chlorophyll as well in plant cells. Again, I just want to point out that there is no ATP directly made here, right? The whole purpose is we're trying to make ATP, but these NADHs are carrying these electrons, and as that electron is being shuttled down its energy gradient, each one of those is an energy transfer, because if we went from oxygen to water, it's just too great of energy. So these are just incremental, small amounts of energy transfer that are manageable by our cell. And what is happening while this is occurring is that hydrogens, protons, H+, are being pumped through the membrane. So here's the same exact thing that we just saw, just another version. So this is a phospholipid bilayer, right? This is just one layer. Let's just take a look at the mitochondria real quick. Where's their picture here? The mitochondria has two membranes. It has an outer membrane and it has an inner membrane. And the spaces that are created between these two membranes and then within this membrane are very different. And they have specific names that you need to note. So the complexes that we're talking about here are in this membrane here. Not the outer membrane, but the inner membrane. So all along this inner membrane here, we have those four complexes over and over and over and over and over again, right? The more the merrier, because the job of this is to make ATP. So these four complexes are all along that membrane, and the portions, the inside of this is the matrix, and then this is the intermembrane space. And do note that, so I'll say that again. This is called the matrix, the innermost component here, and then the darker orange between the two membranes is called the intermembrane space. Inter means between, so that literally means between membrane space, the intermembrane space. So they have different environments. One of them has a higher acidity than the other, and we're going to see how that happens and why that's important right now. So here are those four complexes. They are in that inner membrane. Here is the matrix, the very inner part of the mitochondria, that's yellow. And then the orange here is the intermembrane space. So there will be the outer membrane out here for the mitochondria. And what happens is, as those electrons are being shuttled through this electron, here it is, NADH, which is carrying those electrons, as it's being shuttled through this electron transport chain, through the cytochromes, it is causing hydrogen protons to be pumped in from the matrix to the intermembrane space. And this is creating a gradient. So this area here, the intermembrane space, has a lower pH, meaning it has much more protons, right? Because a lower pH means that it's more acidic. So this darker orange area is very acidic because of this pumping action by all of these cytochromes and all of these complexes that are present in this inner membrane over here. And just so that you see yet another representation, because you never know how you'll see this on an exam, here is a mitochondria diagram. Same thing. So we have the outer membrane and we have another outer membrane, right? So let's just imagine that this whole thing is the mitochondria. Then we have the second membrane, which is the inner membrane. Between the outer and inner membrane is the intermembrane, which just means between membrane space. This space is very acidic. And then in here in the inner mitochondrial matrix, or just the matrix, it's less acidic. In this inner membrane here, this phospholipid bilayer is where we have our complexes. So here's 1, 2, 3, and 4, and they will be all along it. But here's another one, 1, 2, 3, and 4. And this is the electron transport chain. All of the NADH and FADH2s are brought to complex 1 and complex 2, and the electrons are taken from them because these have a higher electronegativity. So the electrons are going to be moving down their energy gradient. Each one of these components is more and more and more and more and more electronegative until the very bottom where we have oxygen hanging out, ready to grab that electron. It's the most electronegative. So it's there kind of like a vacuum, sucking that electron to it. Okay, so I still haven't mentioned how ATP is made, right? We still don't know that. All we know is that we oxidize glucose, we harness those electrons, we shuttled them through the electron transport chain, and in doing so, we've created a gradient, a proton gradient. Okay? Right. So, that part that we just learned was the electron transport chain, right? The shuttling of the electron and the pumping of the protons into this intermembrane space to create a proton gradient, where there are many more protons in this dark orange space than there are in this light orange space. So this next step is where we actually make ATP, and this is called chemiosmosis. Chemiosmosis. Write that down, chemiosmosis. So what actually happens in chemiosmosis? Well, we have a special protein here, an enzyme, called ATP synthase. And that should tell you all you need to know right there, right? ATP synthase. To synthesize something means to make it. And anything that ends in an ase is an enzyme. So this is an enzyme that synthesizes ATP. So make sure you know that enzyme. You will probably see that. So ATP synthase is sitting over here. What ATP synthase does is it allows for protons, right? We created this gradient from pumping all this up. It allows these protons to flow through it. And in doing so, it spins a rotor that's sitting here, which is creating energy, right? This proton motive force, which is what it's called, the proton motive force, spins this rotor, and the rotor pops together an ADP and an inorganic phosphate to make ATP. Let's see if we have a video. So what we see here is an enzyme, ATP synthase, sitting in the inner membrane. Over here you would have had your complexes, 1, 2, 3, and 4. And let's see, here we go. So we had pumped all the hydrogens up here at the top, so they have a higher gradient, and now they're able to move down their concentration gradient. Again, there's no energy being used here. The hydrogens are just falling down their concentration gradient. And in doing so, they're spinning this rotor here. And I just think of like if you go into a major city and you spin one of those doors, it's just like that, right? So it's spinning that rotor, and then in doing so, it's providing energy for this bottom part to place together a ADP and then an inorganic phosphate. and what's coming out is ATP. So that process there is chemiosmosis, just the protons moving down their concentration gradient. So that gradient is called the proton motive force, okay? And that is what is driving the creation of ATP. So another way to say this is that, and it's just written right here for you, but the energy that's stored in the proton gradient across that membrane, where one part of it is more acidic and it has more protons, the energy stored in that gradient couples that redox reaction, the electron transport chain, to ATP synthesis. The electron transport chain created this proton motive force. Again, there's no energy being used here. The electrons were taken off of NADH because of an energetic pull, because all of these are more electronegative. No energy being used here. So this proton motive force is created from that electron transport chain, and then that proton motive force drives the production of ATP. So we say that the proton motive force, it's like the middle step, right, is what's coupling. Coupling just means like attaching, connecting. So this proton motive force, this gradient, couples the electron transport chain with the production of ATP. And again, make sure you know what chemiosmosis is. Chemiosmosis, the energy coupling mechanism. So that is how we create ATP. And here's just one more picture of ATP synthase. Again, make sure you know that enzyme. There's so many enzymes that are involved in our metabolic processes, but there's a couple that you always have to know, and I try to call them out for you. So ATP synthase is definitely one of them, and you're going to see it again in photosynthesis too. So it's good to just know it now. Also, make sure that you know where everything is occurring in the mitochondria, because those you can see pop up on exam questions, just to make sure that you know where everything is occurring. So both of those steps, the electron transport chain and chemiosmosis, are what make up oxidative phosphorylation. So we've gone through all of the steps of cellular respiration. So let's look at this one more time with an accounting of everything that we've created, because again, this is another thing that you need to know, the amounts of what's made. So we started off with glycolysis. We had our 6-carbon glucose. We made 2 3-carbon pyruvates. Out of that, we netted 2 ATP and 2 NADH. The NADH goes over here to go toward the electron transport chain, and ATP is around for the cell to use. Then we shuttled our pyruvate across the mitochondrial membranes. There was a 3-step process there, right? We chopped off one of our carbons and released it as a carbon dioxide. We created an NAD, and we created our 2-carbon acetyl-CoA because we stuck a coenzyme A onto it. And we have 2 of them because we have 2 pyruvates. So each pyruvate makes 1 carbon dioxide, 1 NADH, and 1 acetyl-CoA. So we have 2 of everything. Those acetyl coenzyme A's then get shuttled into the citric acid cycle. Each turn of the citric acid cycle gave us 3 NADH and 1 FADH2 and 1 ATP. We got 2 turns of it, right? So altogether we netted 6 NADH, 2 FADH2s, and 2 ATPs. Okay, so far we've only made 4 ATPs, but we've made a bunch of NAD. Let's see, how many have we made? 2, 4, 6, so 12 NADHs and 4 FADH2s. So note that for each glucose, you make 12 NADHs and 4 FADH2. Then those electron shuttles get sent over to the complexes in the inner membrane of the mitochondria for the electron transport chain. And we create ATP with the electron transport chain and chemiosmosis. And this makes 26 to 28 ATP. So you can see that just 12 NADH and 4 FADH2s can make almost 30 ATP. So this is your bang for your buck here, oxidative phosphorylation. So altogether, we've created anywhere from 30 to 32 ATP per glucose. Make sure that you note that. Per glucose, 30 or 32 ATP. And just so you know, you might also see if I have 1 mole of glucose. Well, remember, a mole is 6.02 times 10 to the 23rd molecules of glucose, but if you have 1 mole of glucose, you'll just make 30 to 32 moles of ATP. So here is the linear snapshot of our energy flow in cellular respiration. We started off with glucose, which needs to be oxidized, so we're going to break it down, and in doing so we release electrons, which get taken up by NAD+, so we make NADH, which then goes to the electron transport chain, which then creates the proton motive force, which created ATP. And remember how I said in the very beginning that most of our energy is lost in the form of heat? So only about 34% of the energy in a glucose molecule actually gets transferred into creating ATP. So the rest of it is lost as heat. So just as a reminder here, what I'm talking about, only 34% of what you take in and eat as energy gets made into ATP. So the rest of it, 60-something percent gets lost as heat. Okay, so that was only one catabolic pathway, right? We're breaking stuff down in catabolic pathways to produce ATP. So that one was cellular respiration, right? We're also going to cover a couple of other ones really quickly. So we have two basic things on here. We have respiration and we have fermentation. And there are two types of respiration: aerobic and anaerobic. Aerobic here means that there's oxygen involved, right? We have the word aero, air. And obviously oxygen is in our air. So aerobic respiration requires the consumption of organic molecules, but because that's what we're breaking down for energy, but specifically, in addition, we need to have oxygen present. Anaerobic, an means without, so without oxygen, anaerobic respiration, again, we're going to be consuming compounds because that's what we eat. Other organisms also need to take in compounds to break them down for energy, but anaerobic respiration doesn't require oxygen. Cellular respiration, which we just went over, included both of these types. We had anaerobic respiration and aerobic. And can you think of where each one was? Let's go look at that right now. So, which of these parts didn't require oxygen and which ones did? There was only oxygen at one part, right? We didn't see oxygen come into the equation until oxidative phosphorylation. So glycolysis is anaerobic. We don't need any oxygen here. This step is anaerobic, and the citric acid cycle is anaerobic. We don't need it. Oxygen here, but oxidative phosphorylation is aerobic. So cellular respiration, this entire process here, included both aerobic and anaerobic components to it. Some organisms and even some of our own cells are able to do another type of catabolic mechanism pathway called fermentation. And I know you've heard of fermentation before. Fermentation is anaerobic. We don't need any oxygen to ferment something. And fermentation is the partial degradation of sugars. So again, we're starting off with sugar as our main organic molecule that we're breaking down. So let's look at all this real quick. Here's a quick snapshot of anaerobic versus aerobic respiration here. So we have anaerobic, right? Here's our glucose. It gets broken down into pyruvate. This is glycolysis. And here is fermentation occurring. We're going to get into this in just a second. But if we do have air present, so there are some organisms that can do both. They're facultative anaerobes or facultative aerobes. So depending on what is present, they could use oxygen or not. We just saw what happens as far as how much ATP is created if you have an oxygen and you want to use it versus if you don't have oxygen, right? No oxygen present, we can only make 4 ATP from this glucose. Oxygen present, we made 30. So if an organism is able to do aerobic respiration, cellular respiration, and there's oxygen present, it's going to choose this. But if there are some organisms that can just do anaerobic respiration, we can do that a little bit obviously we can do that as well. If we have low oxygen circumstances, actually let's get into that real quick. So let's talk about fermentation and anaerobic without oxygen respiration. So again, this is producing energy without any oxygen present. Remember that the oxidative phosphorylation part of cellular respiration, the very end part over here, relies on oxygen pulling the electrons down their energy gradient, right? Oxygen needs to be here in order to be pulling that electron down this electron transport chain. If there is no oxygen present, and that even goes for us who we are cellular respirators, this electron transport chain will cease to exist. That is why we need oxygen. Everyone knows that we use our lungs to take in oxygen from the air. It goes into our blood, but then I find that people usually don't know why we need oxygen, but here it is. This is it. We need oxygen as the electronegative acceptor of electrons at the end of the electron transport chain. So if we have a low oxygen circumstance or there's no oxygen present, the electron transport chain will cease to exist, but we still do have a way of creating some ATP in anaerobic circumstances. So in this case, there are two options, right? We have glycolysis. We always start with glycolysis. So glycolysis can then either couple with anaerobic respiration, or fermentation. Anaerobic respiration uses an electron transport chain with a final electron acceptor that is not oxygen. So it has like a modified type of electron transport chain, this anaerobic respiration. Because remember, there's no oxygen present, so it has to be a little bit different. Fermentation uses a different type of phosphorylation instead of an electron transport chain to generate ATP. Let me just back up a second and go over what this is talking about, substrate level phosphorylation. We saw in glycolysis that we were able to make 4 ATP. And how do we make ATP? We take an ADP and we add a phosphate, right? The same exact thing happened in oxidative phosphorylation with the enzyme ATP synthase, which is here. Same exact thing. To make ATP, you take an ADP and you add a phosphate group onto it to make ATP. This is phosphorylation. Adding a phosphate onto something is called phosphorylation. So we have two ways to phosphorylate to create ATP. We can either have enzymes make it for us, like we see here, right? This is an enzyme, phosphoglycerokinase or pyruvate kinase, right? These are our two enzymes that are phosphorylating the ADP to make ATP. So we can either have enzymes make our ATP phosphorylate for us, or we can have the electron transport chain drive the phosphorylation, right? It created the proton motive force, which drives the turning of this turbine here to create ATP. Those are two different types. Those are substrate level phosphorylation versus oxidative phosphorylation. When you think of substrate, what do you think of? Hopefully you think of an enzyme, right? The substrate is what binds to an enzyme. So substrate level phosphorylation is the use of an enzyme to make ATP. So here it is. Here's an ADP. Here is a substrate with a phosphate on it. The enzyme pops the two together. The ADP gains that phosphate, so we make ATP, and it releases the substrate, the product here, without the phosphate on it. That's exactly what's occurring here. So this is substrate level phosphorylation. The same exact thing happened with the citric acid cycle, right? Here's the ATP being made. Again, this is with an enzyme. You don't see the name of the enzyme here, but this is also substrate level phosphorylation. So glycolysis and the citric acid cycle are both examples of substrate level phosphorylation. Oxidative phosphorylation is powered by redox reactions, and it says here that it generates 90% of the ATP. We know that, right? We have a count of all of the ATP. Almost, 28 are made by oxidative phosphorylation, whereas only 4 are made by substrate level phosphorylation, right? So, bringing it back to our talk on fermentation. Whoops. Anaerobic respiration is going to use a modified electron transport chain to kind of mimic oxidative phosphorylation, although it's not oxidative because there's no oxygen, right? It's anaerobic. And fermentation is going to use substrate level phosphorylation. So we're going to be using enzymes instead of electron transport chain to generate ATP. So looking at fermentation, we actually have two types of fermentation that we're going to talk about. Again, fermentation, we start off with glycolysis. Right? Glucose into pyruvate. And then we have two alternative ways that we can go from there. So these are our two types of fermentation. We have alcohol fermentation and lactic acid fermentation. So alcohol fermentation is, after glycolysis, we formed pyruvate, right? So we're starting with pyruvate for fermentation. So for alcohol fermentation, that pyruvate gets converted to ethanol, right? This is how our alcoholic beverages are formed. Alcohol fermentation is forming the molecule ethanol. So let's look at that. So in the first step, we have a release of carbon dioxide, and in the second step, we produce NAD+ and ethanol. So let's see this. So here we have both types of fermentation. Let's focus on alcohol fermentation on the left. So again, we start with glucose, goes through glycolysis, and we make 2 molecules of our 3-carbon pyruvate. Again, glycolysis makes 2 net ATP. Same here. This pyruvate, our 3-carbon pyruvate, gets broken down by another enzyme. It causes the release of one of the carbons and 2 of these oxygens. So we lose a carbon dioxide here and we create a new 2-carbon molecule, acetaldehyde, and we create 2 of those, right? Because we have 2 pyruvates, each one of them gets broken down. So we have 2 acetaldehydes. Now, acetaldehyde, this 2-carbon molecule here, is going to be transformed into the molecule ethanol. Now, this is a very important cycle right here. This is the NAD plus to NADH to NAD plus. This is an NAD plus regeneration cycle. What happened when we made ATP from glycolysis here? When we took our glucose and turned it into pyruvate, we created 2 NADHs, right? We harnessed a couple of electrons from this glucose that are now being held in this shuttle. Now, remember, we are not going through cellular respiration here. We're not going to be taking this into any mitochondria. There's no oxygen present, so this can't go through any sort of electron shuttle, electron transport chain. So instead, the NADH that we generate from glycolysis is used in this step here to turn the acetaldehyde into ethanol. So the NADH carries electrons, and those electrons and a couple of hydrogens get transferred onto this molecule, which becomes ethanol. And in doing so, we regenerate NAD+. So that means this molecule can go back to new glucoses that are coming into the cell and being broken down via glycolysis, and now we have a reusable cycle here, and with the NAD plus regeneration, and that's very important. So that is alcohol fermentation, right? We begin with glycolysis, and then there is an NAD plus regeneration cycle here, and pyruvate gets turned into a new molecule, acetaldehyde, which then gets turned into ethanol. The second type of fermentation is lactic acid fermentation. In lactic acid fermentation, pyruvate is reduced by NADH, just like it was in alcohol fermentation, and we reform the NAD+, but we have new end products and no release of carbon dioxide. So let's look at that comparison here. So here's the lactic acid fermentation. So again, we're going to begin with glucose being broken down into pyruvate via glycolysis. We create a couple of ATP and we created 2 NADHs. Same thing as always. Now, there is no release of carbon dioxide here. So pyruvate is maintaining all 3 carbons. This NADH, the electron shuttle, is able to give some electrons to here as well as some hydrogens that become associated, and we have a new molecule that's formed, which is lactate, also known as lactic acid. And again, lactic acid is a 1,2,3 carbon molecule. Okay, so we see that they're very similar. They both have this NADH, NAD plus cycle, and NAD plus is regenerated. If NAD plus was not regenerated here, again, there's no oxygen present, so any sugar that would go into these cells would be broken down via glycolysis. Any of these coenzyme NAD pluses which are present would be grabbing these electrons, and if there was nowhere for these to go, and there isn't if there's no oxygen present, there's no electron transport chain, and there's no oxidative phosphorylation. So if this cycle didn't exist, then these molecules would remain as NADH and would never be regenerated. if they weren't regenerated, then the cell would cease to be able to break down more glucose and make more ATP, right? So in doing this, we are regenerating this molecule, which can allow the cell to continue to make ATP. So this is a very important part of it, this NAD plus regeneration. And just one more time, kind of looking across the board at all of them. Here is our anaerobic circumstances here. Anaerobic conditions versus aerobic. So glycolysis is anaerobic. We don't need oxygen for glycolysis, and nothing does. Glycolysis is a very old metabolic pathway. It's in single-celled organisms. It's in bacteria, right? So it's been around a very long time. Bacteria are 3.5 billion years old. They came before us. So once glycolysis occurs and we have pyruvate, then the cell, depending on the type of cell also, so yeast fermentation is alcohol fermentation. So that's why it creates some carbon dioxide and it creates the molecule ethanol, which, so, you know, to make beer and wine and all of that, right? This is the type of fermentation that would occur. Lactic acid fermentation, on the other hand, we actually are able to do that. So let's go back to this slide here. Sorry, here we go. So who can do some lactic acid fermentation? Fungi and bacteria to make cheese and yogurt. And then for us, our muscles are able to utilize lactic acid fermentation when we have low oxygen conditions. So this happens during strenuous exercise. Obviously, the whole point of this, again, is to make ATP, right? We need to be able to regenerate this NAD plus. Sorry, here we are. NAD plus over here. So if we didn't go through lactic acid fermentation, then our muscle cells would go through glycolysis and then kind of stop. If there's a low oxygen, if we're exercising a lot and we're just not getting a lot of oxygen, or we're depleting it a little bit. So if you've ever heard of when you're sore, it might be due to lactic acid buildup in your muscles, and you wonder, have you ever wondered, well, where does that come from? Here it is. After strenuous exercise, if you had low oxygen conditions, your muscles may have kicked into lactic acid fermentation, so that's why you have some lactate built up in your muscles. And to continue the comparison, so comparing fermentation with anaerobic and aerobic respiration, all three of them used glycolysis, right? Glycolysis was the same throughout. It's always in the cytoplasm. It's always anaerobic. It always nets 2 ATP and it created 2 NADHs. It oxidizes glucose to harvest all of that chemical energy. In all three, NAD plus is an oxidizing agent, right? It is what accepts the electrons and becomes reduced to NADH. And you might see that on an exam that way. What's the reduced form? NAD plus or NADH? Reduced means it gained an electron. So just remember that NADH would be 2 electrons. That's the reduced form. NAD plus is the oxidized form. In fermentation, an organic molecule acts as the final electron acceptor. So in this case, pyruvate is the electron acceptor and becomes lactic acid. Acetaldehyde is the electron acceptor here and becomes ethanol. Those are for fermentation, whereas for cellular respiration, of course, the final electron acceptor is oxygen, right? In cellular respiration, the electrons are transferred through the electron transport chain, and cellular respiration produces way more ATP than fermentation, right? Fermentation only has glucose creating ATP and there's only a net of 2, whereas cellular respiration, we have oxidative phosphorylation, which makes 28 extra ATP. So there's a big difference in energy creation by them as well. I mentioned obligate anaerobes before. I may have actually misspoke, but obligate anaerobes use fermentation and or anaerobic respiration, but cannot survive in the presence of oxygen. So obligate means, to be obligated to something. So they're obligate anaerobes. So they do not want to be in the presence of oxygen. So they will not be able to go through aerobic respiration, only fermentation or anaerobic respiration. And then we have some organisms that are facultative anaerobes, such as yeast and bacteria. So these organisms can survive using either fermentation, which is anaerobic, or cellular respiration. So they'll just do what's available to them. Those are facultative anaerobes. For facultative anaerobes, this pyruvate here is a fork in the road of one of two ways that they can go. So they go through glycolysis. If there is oxygen present, they're gonna go through cellular respiration because, again, you get way more bang for your buck here. If there is no oxygen present, they are still able to survive via some form of fermentation, whether it's ethanol or lactic acid fermentation. So facultative anaerobes are able to make the best of whatever situation they are put in. All right, so that was a lot that we've gone through. We did the citric acid cycle. We talked about fermentation, right? This is a major metabolic pathway. So to finish this chapter, we're just going to kind of take a step back and look at how glycolysis is connected to other major metabolic pathways in the body. I'm not sure if you can really make out all of the words on this, but this is a human metabolism map. As you can see, it's broken up into the 4 macromolecules that we learned in chapter 5, right? We have carbohydrates, lipids, proteins, and DNA and RNA, which are nucleic acids. So you can see that they're all connected, right? And we learned a very simplified version, right? Glucose is the beginning of glycolysis and gets broken down to pyruvate, and then it goes into the citric acid cycle. Well, here's the citric acid cycle. It's at the heart of all of this. And you can see how there's different inlets and outlets, different ways that products can feed into this system, right? It's not just a one entry type of a system. So let's see if you can see this. Here's glucose, right? Glucose, and here's pyruvate. So here's glycolysis right here. So you can even see that there are other aspects. Here's lipids. When you break down fats, they break down into different components and they can feed into glycolysis at different parts of this pathway here. Even the carbohydrates. So we have glycogen, we have starch, we have sucrose, right, which is table sugar. We have fructose, which is in fruit. So they all can feed into this this pathway at different times. So I just don't want to give the impression that there's just one way in, one way out. It's a very complex, intertwined metabolic scaffold here. That breakdown of lipids that I just showed you that can feed into glycolysis, the breakdown of fatty acids is called beta oxidation, and you should know that. So glycolysis was the breakdown of sugars. Beta oxidation is the breakdown of fatty acids. And remember, fats, triglycerides, are composed of a glycerol backbone and fatty acids, right? So this is a sugar and these are fatty acids. So the fatty acids need to be broken down via beta oxidation. Here's a simplified version of that metabolic map that I just showed you. So you see at the heart of it, we have glycolysis, glucose into pyruvate, we make acetyl-CoA. Here's our citric acid cycle. Here's oxidative phosphorylation. Here's fats feeding in. The glycerol is a carb. It's an alcohol actually, and it feeds in right here into glycolysis, and then the fatty acids get broken down via beta oxidation. And beta oxidation basically just chops up fatty acids into chunks of 2 carbon molecules. So perfect acetyl is a 2 carbon molecule, right? So it kind of gets chopped up and broken down and made into acetyls, and then the CoA gets added onto it. So it's perfectly goes right into there. And then proteins are made of amino acids, and they get broken up into various there's 20 different amino acids, right? So depending on what's on them, they can be chopped up, and then they enter into the cycle in various spots as well. Now, we really can't have a lecture without touching on some feedback loop mechanisms, right? We've kind of we learned what a negative feedback loop was in the very beginning of the class, and now as we go through, we're learning examples of regulation and feedback. So we're going to learn about one particular enzyme, which is used for metabolic control, particularly control of catabolism. So let's see. The enzyme is called phosphofructokinase. It's also called PFK, capital P, capital F, capital K, phosphofructokinase. So when we looked at glycolysis and we looked at the steps in glycolysis, one of those steps, I believe it's the third or so, is uses the enzyme phosphofructokinase. It turns this fructose 6-phosphate into fructose 1,6-bisphosphate. You can go back and take a look at it. Actually, I guess I can just pull it up real quick. Boom, right here. Third step, phosphofructokinase. So this enzyme is taking ATP, and this is substrate level phosphorylation. We are phosphorylating this molecule here to create fructose 1,6-bisphosphate. Bi means 2, so there's 2 phosphates on here. So this enzyme is a major regulation point. It starts or stops this process, right? We always need regulation in our pathways. So here we go, phosphofructokinase. So what starts and stops it? Well, the purpose of glycolysis is what? We're trying to make ATP. So that's the end goal. Usually the end goal is the thing that will inhibit this, right? So if we have created a ton of energy, we ate, we broke down all of our sugar via glycolysis and went through all of our systems, and we've created a ton of ATP. We don't need to keep creating it. So ATP itself is an inhibitor of this enzyme, phosphofructokinase. What else is an inhibitor? We see citrate. Recall that citrate is the 6-carbon molecule that we created in the citric acid cycle. We took our 2-carbon acetyl-CoA, we added it to our 4-carbon oxaloacetate, and we created a 6-carbon citrate. So this is just once again another product to let us know that we are going through this system. And if we have a ton of citrate, we're going to have a ton of NADH and ATP, and it's going to be going through phosphorylation. So this also lets us know that we are well on our way to making a ton of energy. So of course that's also going to be a good inhibitor to tell us to stop. We don't need any more. So ATP and citrate are both inhibitors of phosphofructokinase. but what will stimulate it? So that means if we are lacking energy, if we are lacking ATP, well, what happens when we make ATP? We use it, right? We use it, and how do we use it? We break it apart. We cleave off one of the phosphate groups to make ADP, and then we'll actually cleave off another one to make AMP. So we have ATP, adenosine triphosphate. We clip one off, it's adenosine diphosphate. We clip one more off, it's adenosine monophosphate. So if we have a ton of adenosine monophosphate hanging around the cell, that lets us know that we're clipping our phosphates off of these. We're running low on ATP. We have used them all and we need to make more. So this is going to stimulate phosphofructokinase so that we go back through glycolysis so we can continue making ATP. So that is a perfect example of a feedback mechanism. And here is just another picture of phosphofructokinase PFK1, because there's another one, PFK2. And that will wrap it up for this chapter. So yeah, there's a lot in this chapter. And I know this is a little bit of a long video, but it needed to be this long because there's just so much to it. So make sure that you focus on the things that I mentioned as far as these steps. You want to know the input and output, the reactant and the product. You want to know how many NADH and ATP are created. You want to know where this is occurring. What else? It's really important to understand in oxidative phosphorylation that there's these two components, the electron transport chain, which doesn't use any energy, but it creates this proton motive force, and then the chemiosmosis, which uses that proton motive force to drive the production of ATP. Again, no energy is made here, used here. No energy is used here, and we're making ATP. This whole thing is oxidative phosphorylation. In the mitochondria, make sure you know the names, the outer membrane, the inner membrane, the intermembrane space, the matrix, or the inner mitochondrial matrix as it's called. Make sure you know where these complexes are located. They're on the inner mitochondrial membrane. Cytochromes, know what those are, right? For lactic acid, we have our two types of lactic acid. For fermentation, we have two types of fermentation, lactic acid and alcohol. They're both anaerobic. They have that really important component where they have an NAD plus regenerator, because otherwise we would break down glucose, make NADH, and then nothing's there to restore this cycle. So that's why fermentation is really important. And then of course phosphofructokinase as a regulatory spot. So hope that clarified some things for you, and the next chapter is going to be photosynthesis. Good luck.